Today I will be taking on 1 hour extra session for the NPTEL course and genetic engineering theory and application. So mostly we had covered all the topics that were covered in the lectures. We went through the different concepts of genetic engineering, the upstream processes and the downstream processes. So now in this session, one topic which was remaining was application of genetic engineering or how biotechnology is used in social welfare. That topic will be covered today. So let's start today's session. So other than that, if any one of you have any doubt, then we can take up those questions as well. Okay, good evening and welcome to the extra session on this NPTEL course, Genetic Engineering Theory and Application. I'm Akash Jyoti Pathak and I'm one of the PMRFTAs for this NPTEL course. And this course is being run by Professor Vishal Trivedi of IIT Guwahati. This topic was actually covered in week 11. Today we will look at some of the applications of genetic engineering. So in the last class, what were we discussing? So can anyone summarize what are the different applications of genetic engineering? So we must all of you have read in class 12 as well in basic biology. We have read about the how genetic engineering is useful for generation of genetically modified crops for synthesis of therapeutic proteins or uh, you must be knowing about the example of humulin or the recombinant which is basically recombinant insulin. So there are different applications of genetic engineering. So today we will learn about some of them. So the first application is gene therapy. How is genetic engineering in useful in gene therapy? So what is basically gene therapy? It is the production and supply of recombinant proteins. So the production and supply of recombinant proteins is a temporary solution for the treatment of a disease or condition. So basically, as you know the example of insulin. So when a person is not able to synthesize its own or the cells of the pancreas, they are not able to produce insulin, then the patient is given insulin injection. To so that is an example of how recombinant proteins can be a solution for treatment of a disease condition. But that is not a permanent solution. Why is it so? Because the patient has to be supplied with the recombinant insulin. There are doses, there are different types of insulin, and each type of insulin has a particular dose. Some has to be given every day, some have to be given like once a week. The gene therapy is a method which can provide a permanent solution in the sense that if we can modify the gene or we can through the process of genetic engineering, um, if we can the mutated gene which is there can be replaced with a correct gene to in a diseased individual so that the individual can individual does not have to survive on the on drugs or on these regular treatment of these insulin. So that is what is gene therapy. So in another approach, human expression system is used to produce the proteinaceous factor after inserting the recombinant clone into human cells or inside the human body. So the recombinant DNA is packed into the appropriate DNA delivery system. So there are different DNA delivery system. Either it can be a virus mediated DNA delivery system or a liposome mediated DNA delivery system. So that what happens, so once you encapsulate the gene within a virus or within a liposome, that is then uptaken by into inside the cell by the process of endocytosis, and then that gene that is present, that is through the process of homologous recombination, there can be the mutated gene can be swapped with the correct gene. So there are different mechanisms or details of how we can basically change the mutated gene with the correct copy of the gene. So that is basically gene therapy, where you are changing the genetic makeup of a person, or you are trying to correct the altered gene which is causing the disease. Okay, so to correct the mutated gene or encode a therapeutic protein drug to provide treatment. Okay, so or other thing can be that you can if you can insert a gene which can code for the therapeutic protein inside the body, then that person does not have to take the medicine, or does not have to take that insulin injections at regular intervals of time. Okay. That is, so there can be two types of gene therapy. One is somatic gene therapy and is germline gene therapy. So in somatic means what? In the mitotic cells. In the if we are inserting the gene in the mitotic cells, then that is called the somatic gene therapy. So in this therapeutic approach, the therapeutic genes is transferred into the somatic cells as per the requirement of individual to treat the functional defects. So this treatment does not move to the patient's offspring or next generation. So basically what is done is the correct copy of the gene is inserted into the mainly the bone marrow or the that can divide continuously. So that is somatic gene therapy. And another is the germline gene therapy, where you are where the germ cells are made to or they are transformed by the introduction of the required gene to produce the protein of the or correct the mutated gene. So what will happen when something is inserted into the germ cells, then that particular gene will be transferred to the offspring as well, isn't it? So this allows the treatment move to the patient's offspring or next generations. So although the approach seems promising in providing long-term solution to treat genetic disorder, but there are several ethical, technical reasons and possible future risks. So as a result, this gene therapy, there are a lot of research going in this area, but still now there are very few diseases which can be treated with this gene therapy based treatment. So what are the problems that are associated with it? So the main problems are that it is short-lived, isn't it? So the therapeutic gene delivery into the cells gives short-term effects either by rejection or recombinant DNA or suppression of the gene expression. Due to this problem, patient needs to go for several rounds of gene therapy. And the third can be the immune reaction. So, virus containing genes is treated as a foreign object, and the immune system is stimulated to attack the invader. So, when you are doing a virus mediated gene delivery, then often the virus is chosen that is not immunogenic, but sometimes these things can go wrong, and the virus can. Can activate the immune system as well of the host. So the viral vector used as a vector to deliver the gene causes much adverse immune reactions and toxicity in patients. And also there can be disturbance of the host physiology. So if the gene, when you have synthesized this gene and then delivered into the patients, then that particular gene, whether it will be correctly integrated at the right position, or whether um, the correct homologous recombination will take place. So these things are so sometimes they can cause they can be integrated in such a place that it can activate a oncogene, and that can cause cancer as well. So if the gene integrate into a wrong place in the genome, it may cause functional defects. In few cases, it may disrupt the function of the it may disrupt the function of the tumor suppression genes, resulting in the development of tumor. For example, X-linked severe combined immunodeficiency, which is kid injected with the transformed hematopoietic stem cells, lead to the development of T-cell leukemia. Okay, so this X-linked severe combined immunodeficiency. So these patients have a form of immunodeficiency, and the patients were injected with transformed hematopoietic stem cells, but that led to the development of T-cell leukemia. Okay, so there are several challenges of gene therapy, or there's some certain problems that are associated with gene therapy. So that is why this method of treatment is not yet that universally um, or there are not many treatments that can or basically gene therapy is the last line of treatment even in cancer or in for different diseases. So a person with hereditary disease can be cured with the help of what? So answer this question. So just now we discussed that if it is a hereditary disease, then the only so if it is a hereditary disease, then so if you read the options, yoga, dialysis, chemotherapy, gene therapy, then you can yeah, then you will understand that the correct option is option A, gene therapy. Isn't it? Because chemotherapy is for cancer patients, dialysis is when for kidney failure patients, yoga, and these are not for curing the disease, but so the correct answer is option A, gene therapy, where you can replace the mutated gene with the correct gene. Okay. Now let's understand about this hybridoma technology. So hybridoma technology is a method where you can synthesize monoclonal antibodies. Okay. So how does this technology work? So basically, myeloma cell is is a cancer cell basically, which can divide indefinitely. So a myeloma cell is fused with a spleen cell, or so the spleen basically the cell that is produced, that is called a hybridoma. So it is a hybrid cell which is having the property of uh, both the myeloma cell, in the sense that it can divide continuously, and also it has the property of a spleen cell. So the spleen cell can produce antibody. So that is hybridoma technology. So hybridoma technology uses when you are generating a hybridoma using this technology, there can be three types of cells that are produced. One is the hybridoma. So hybridoma cell or the hybrid cell is the one which is the cell which is formed by the fusion of the myeloma cell and the spleen cell. Okay, so what is desired is that this technology gives you only the hybridoma cell, but during this process there are other cells also that are produced. So there can be unfused spleen cell, and there can be unfused myeloma cell. So you do not want the unfused spleen cell and the unfused myeloma cell. Uh, for myeloma cell in this in vitro, when you are synthesizing this and synthesizing the or producing antibody by this technique, monoclonal antibody. So how do you select for the hybrid cells? So that selection is done by this method, HAT selection. So it is HAT selection is basically, so there are three types of HAT. What does HAT stand for? HAT stands for hypoxanthine aminopterin thymidine selection. Okay, thymidine selection. Basically, your media has these three components: hypoxanthine, aminopterin, and thymidine, which will select only those cells which are only the fused cells, only the myeloma and the spleen fused cells. So this is a classical method used to isolate hybridoma cells for monoclonal antibody production. This principle relies on blocking one DNA synthesis pathway and forcing cells to use another, which only hybrid cells can do. So what it is doing is basically this hat selection is allows you to select only the fused cells, only the myeloma and uh, the uh, la- the fused spleen cells. Okay, basically when what happens is the DNA that is synthesized in the cell through two pathways. One is the salvage pathway and another is the de novo pathway. So we will understand how these three components in the media forces the cell to use only one of the DNA synthesis pathway. So what is the principle of hat selection? So hat medium contains three components. One is hypoxanthine, second is aminopterin, and third is thymidine. So what does aminopterin do? Aminopterin blocks the de novo pathway of DNA synthesis. So the cell must use the salvage pathway to make DNA. The salvage now so your aminopterin, the function of aminopterin is to block the de novo pathway for DNA synthesis. Okay, and so that now once this de novo pathway is blocked, then the DNA synthesis will take place by the salvage pathway. Now the salvage pathway requires two enzymes. One is AGPRT and another is TK from myeloma cells. Now, what will happen? What is done is when the for synthesis of monoclonal or generation of monoclonal antibodies, only the myeloma cells which are having a mutation in this AGPRT gene are selected. So myeloma cells used in this process are mutated so that they lack this particular gene which codes for this enzyme, making them unable to use the salvage pathway and thus they die in the hat media. So for salvage pathway, you require these two enzymes, CGPRT and thymidine kinase. So now since these B cells have a mutation in this AGPRT, so the unfused B cells, they will die in this media, in this hat media, and the because the now the de novo pathway is also not running and the salvage pathway is also not running. Okay, but normal B cells which have AGPRT, normal B cells will have AGPRT, but they will lack but they are not, but they cannot divide indefinitely. So they will also die after some rounds of division. Okay, now hybridoma cells, which are fusion of B cell and myeloma cells, have both immortality from myeloma and AGPRT from B cell. So they survive and grow in the hat media. Okay. So why only the hybridomas survive? So this is because only the fused cells, which are the hybridomas, inherit both the ability to divide forever and the enzyme needed to use the salvage pathway. Okay. So these hybridoma cells, they have the property of both the B cells and the myeloma cells. Now B cells lack the functional EGPRT. Okay, so as a result, they can synthesize their own DNA because the salvage pathway is now going on, and also they have the ability to divide indefinitely because of the property. They are getting that property from the myeloma cells, and so only these few cells are able to survive, while the unfused cells be they will not survive in this media. Okay, and in this way, you can select for only the fused cells. Now, unfused myeloma cells die because they can't make DNA by either pathway, and unfused B cells will die because they are basically mortal cells. They will not they will die after some after or after a few rounds of cell division. Okay, so this selection ensures that only hybridomas which produce the desired monoclonal antibodies survive and can be isolated for further use. So this was explained in the lecture as well. So you see here, you take a mice And then from the mice, you isolate these myeloma cells. So maybe a mice which has been induced or induced for cancer. So from the cancer-induced mice, you are taking out the myeloma cells, and from the spleen you isolate the B cells. Now you fuse the myeloma cells and B cells. So so during this fusion, there will be three types of cells that will be produced. One is the unfused myeloma cells, one will be the unfused B cells. So there will be a few unfused myeloma cells and unfused B cells, and other than that, you will also get fused myeloma and B cells, which are called the hybridoma cells. Now we are selecting the hybridoma cells by growing them in this hat media. Okay, so what are antibodies? So antibodies are basically, they are used in adjuvant therapy to remove the infectious agents or toxic substances. So antibody is antigen. They will act against the antigen, and they will elicit an immune response so that the foreign agent can either be phagocytosed or they can be neutralized, basically. So monoclonal antibodies are generated by the hybridoma technology. So you can see here the monoclonal antibody cell production. So first what you have to do, so first there will be isolation of B cells from the spleen. So then this B cells will be fused with the myeloma, and then you have to screen for the fused cell. So before the isolation of B cells, the mice is immunized. So the mice is given an injection of the, or it is injected with that particular antigen so that it will now start producing antibodies, or it will, uh, then you will test the testing antibody generation, bleeding, collection of serum, and then test for the antibody. And then from that mice, you will isolate the B cells and the myeloma cells, and then you can screen for the hybridoma cells. Step 2 is isolation of B cells. So splenocytes are isolated from the spleen of mice. Step 3, cultivation of myeloma cells. Myeloma cells are grown in the serum containing the complete media. Step 4 will be, so once you have isolated the B cells and the myeloma cells, the next step is fusion of the myeloma cells with the B cells. So how will you fuse the myeloma cells and the B cells? By treatment with polyethylene glycol or the Sendai virus. So this polyethylene glycol will result in fusion of the B cell and myeloma cells. And then you have to screen for the fused cells. So the screening of the fused cells are done by growing them in the hat medium. So hat medium is hypoxanthine, aminopterin, thymidine media for an extended period. So myeloma cells lack salvage. So you have to understand this point. So myeloma cells lack the salvage pathway, and they die in the presence of aminopterin. So aminopterin is basically, what is the function of aminopterin? Aminopterin is inhibiting the de novo pathway. Aminopterin is inhibiting the de novo pathway, and it is inducing the cell to start the salvage pathway. Now for salvage pathway, you require HGPRT, but these myeloma cells lack the HGPRT, or they have a mutation in the HGPRT gene, so that they cannot synthesize the functional HGPRT. So the unfused myeloma cells, when they're grown in the hat media, they will die. Okay, but the fused cells are having the property of both the myeloma cells and the B cells. So the B cells have the functional AGPRT, so they are only able to survive, while the unfused B cells, and so that is what is explained here. So fused hybridomas are screened to test the production of desirable antibody against the antigen. So once you have screened for the fused hybridoma, then then the antibody which will be produced by this hybridoma, that has to be screened, that whether your antibody is able to act against that particular antigen. So monoclonal antibody means that that antibody is specific only against a particular epitope which is present on the antigen. So you have to screen whether that antibody is specific against that antigen by doing some ELISA based assay. Okay. So ELISA enzyme linked immunosorbent assay. So basically, this is one technique which we have discussed in earlier classes. So by this technique, you will know whether your antibody has that affinity against the particular antigen against which it is desired. The step number 6 is multiplication. So the hybridoma is producing desirable antibody. So first step is to have to screen whether whether screen for the fused hybridoma cells. So that is the first step. Then the next step is to screen for the hybridoma cells which are producing your desired antibody. So that will be screened by doing an ELISA. Okay, and after you have screened for for those hybridoma cells which are producing the desired antibody, now you can grow those particular hybridoma in a medium containing IL-6 to establish the hybridoma cell lines. Okay, so there are multiple steps in this process, and it requires very careful. Uh, you need to perform all these steps very in a very controlled and careful manner. Okay, so the hybridomas are transferred into large culture flasks to secrete antibodies. The culture supernatant can be collected and stored at -20. So so basically these hybridoma cells, they will secrete the antibodies in the media. So you will get all your antibodies in the media. So you can directly collect the supernatant and store at -20 degrees Celsius or other lower temperature for future usage. So now answer this question. In monoclonal antibody technology, tumor cells that can replicate endlessly are fused. So tumor cells, which are basically the myeloma cells, are fused with mammalian cells that produce an antibody. The result of this fusion is what? So the result of the fusion of a myeloma cell and a B cell is, that type of cell is called a hybridoma. Okay. So answer is option B. Okay. So now in human, B cells and T cells are matured in. So this is a very basic immunology question. So B cells are matured in the bone marrow and T cells are matured in the thymus. Okay. So that is also clear. Now coming to vaccines. So what are vaccines? So vaccines are basically antigens or pathogen that have been weakened, or they can be either weakened pathogen, they can be dead pathogen, uh, or a live attenuated pathogen, which when injected into the body of an individual, what will it do? It will trigger the immune system of an individual so that the antibodies can be made against that particular antigen. Okay, so vaccine is given to develop immunity against the disease in the human or other vertebrate animals. So we have all received vaccines. So they are the pathogen itself, which has been either weakened or they have been attenuated by treatment with certain chemicals, or they can be a part of the pathogen, which is called the antigen. So there are different strategies to enhance the immunological response to give long-lasting protection against the disease with minimum adverse effect. Okay, so there can be different types of vaccine. There can be killed, attenuated, toxoid, subunit, or conjugate. So what are killed types of vaccine? So in this vaccine, basically you will kill the pathogen and then inject it into the patient, okay, or individual. So in this vaccine preparation, the pathogenic organism is killed by chemical or UV treatment and then used as an immunogen. So it is mixed with the adjuvant to enhance immunological responses and long memory. So what is an adjuvant? So adjuvant is a substance which can enhance the uh, immune system, or which can enhance the activation of the immune system. So the pathogen can either be in killed form, they can be in the attenuated form. Attenuated means when the organism is treated with a chemical to destroy its ability to cause disease. So the pathogen will not be able to cause disease, but it can induce the immune response response within the individual. So as a result, the organism grows and gives stimulation of the immune system for long-term immunological memory. What is toxoid? Toxoid is anything oid means it is toxin-like. So inactivated toxic compounds are used as the immunogen. So that is toxoid. Okay. Now there is fourth is what? Subunit. Subunit is basically so like in the COVID infection, what was that type of vaccine that was used? So either only the DNA, only the DNA which codes for this only a part of the subunit of the virus that was delivered into the patient. Okay, so in this vaccine preparation, a pure protein or antigen is given as an immunogen. So this is the safest form of vaccine. So you're not giving the whole pathogen. The whole pathogen or the weakened form of pathogen is not being used, but a part of the pathogen. Either it can be a part of the for a part of the capsid protein, or it can be or the spike protein on the or say for example on the SARS-CoV-2 virus. So the DNA which codes for the spike protein that was encapsulated um, and uh, and that was injected into the patients. So that particular gene DNA when it codes for that spike protein, it will trigger the immune response and the antibodies will be produced so that this antibodies when they will be stored uh, as a memory, the memory B cells can then produce the antibody against that particular pathogen in case of a real infection. Now another can be the conjugate. So in this vaccine preparation, bacterial coat is tagged with the immunogenic protein to induce production of immune response against the bacterial coat. Okay, so in conjugate type of vaccine, what you're doing, the coat, bacterial coat, say the calyx or the uh, part of the portion of the bacterial morphology or the bacteria is coated with the immunogenic protein. Okay, immunogenic protein means when you are coating the bacteria with that particular protein, it will result in the activation of the immune response when this bacteria will infect the individual. Okay. So answer this question. So vaccines protect us from getting infection by so destroying the infective agent in environment. So vaccines are not doing that. They are not antibodies or so antibodies they cannot antibodies will also not destroy the infective agent. They will neutralize or they will prevent that particular antigen from binding to the receptor. So answer is not option A. Okay. Killing the insect vectors responsible for spread of infection. So the no vaccine has no such property of killing the insect vectors, increasing the immunity of the host. So answer is option C. Basically, you are giving a weakened or attenuated form or a part of the antigen so that the you can activate the body's immune system and as a result what will happen when the live or the virulent form of that pathogen infects the individual, the body can produce antibody or the person will not develop or will not have the infection or it can neutralize the pathogen or destroy the pathogen by the production of antibodies. Okay, so that is the principle of vaccine. Now, option D is protecting the insider. These are all wrong options. Correct option is option C. Okay, so the vaccine used to prevent tuberculosis is what? So we all know so this is a fact-based question. So BCG is the vaccine which is used to prevent tuberculosis. There are different types of vaccine which are given to individuals against different to prevent different diseases. So BCG vaccine, then there are polio virus vaccine. Then there are different. So MMR is again mumps, measles, rubella. DPT is against diphtheria, pertussis, tetanus. These are combination vaccines. Okay. So BCG, so some of the names of the vaccine, if you remember, that is good. So vaccine used to prevent tuberculosis is BCG. Okay. So now another thing is how uh, how we can use genetic engineering or application of biotechnology is for herbicide control. So what are herbicides basically? So herbicides are weeds. Herbicides are weeds or invasive plants that do not allow the growth um of a particular crop. So those are herbicides. So they are basically weeds. So what do weeds grow? They will grow very fast and they compete for the nutrition. So all the they will utilize all the nutrients, the fertilizers which are present in the soil, and as a result, it will not allow the nutrient to be utilized by the plant. Okay, so there can be so herbicides are used in the agriculture to eradicate weeds from the field. Okay, so herbicides are so so whenever a plant is growing, so along with the plant, there are there are weeds also that grows where that utilizes the nutrients from the soil, and they outgrow the plant and inhibit the growth of the plant. So for the inhibition of or to stop the growth of the weed, herbicides are applied. Okay, so now the concern is that the herbicides which are applied, which they should not. So herbicides are they are basically chemicals which are used in agriculture to eradicate weeds from the field. So if the weeds need to be removed from the crop, the herbicide should do little or no effect on the crop plant. Okay, so what is desired that when you are applying this herbicide, it only damages the or it stops the growth of the weed, but it does not affect the main crop, okay, or the plant. So it should have no effect on the crop plants. So hybrid herbicides are either selective towards a class of plant or non-selective to kill all plants they apply to and use more often to kill all vegetation. So sometimes so we have to use a herbicide which is selective against a particular type of plant so that that particular weed can be eliminated, but your main crop remains is not hampered or is not affected. Okay, so what is so how we can develop herbicide resistant plants basically. So one of the herbicide which was for which was first herbicide designed was glyphosate. So glyphosate is one of the first herbicide which was designed to kill weeds. Okay, so now how does glyphosate work? So glyphosate interferes biosynthesis of aromatic amino acids, tyrosine, phenylalanine, and tryptophan by inhibiting enzyme 5-enol pyruvyl shikimate 3-phosphate synthase. So this chemical which is glyphosate, this inhibits this particular enzyme EPSP synthase. So once this enzyme is inhibited, the synthesis of the aromatic amino acid residues. So there are three aromatic amino acid residues, tryptophan, tyrosine, phenylalanine. So the synthesis of these three amino acids are blocked. Okay. Now, when you so what will happen? So when you apply these type of herbicide, then this glyphosate will not only inhibit the growth of the weeds, but it will also inhibit the growth of the plant because it is basically inhibiting the synthesis of these essential amino acids. Okay, so the enzyme catalyzes the so okay, so the treated plant cannot be will not be able to produce these amino acids as well as proteins needed and dyes. So because these three amino acids are present in almost in most of the proteins. So once the synthesis of these amino acids are inhibited, there will be the synthesis of the proteins which are required for the growth, development, metabolism of the plant. They will also be impaired. So the treated plant can will not be able to. So any plant you take, if you treat with glyphosate, then what will happen? That plant will not grow. It will stop the growth of that plant. So you have to engineer herbicide resistant plants. So plants which are resistant to this particular herbicide, so that the weed which is growing, that can the growth of that weed can be inhibited, while the main plant or main crop can grow well. Okay, so there are two approaches how you can develop such herbicide resistant plant. So how are herbicide resistant plant engineered? So first is what you can produce a genetically modified crop plant that is designed in a manner that it has an alternate pathway to supply the aromatic amino acid to compensate the inhibition of EPSP. So you can engineer a herbicide resistant plant which other than this EPSP synthase, it has an alternate pathway by which it can synthesize the three amino acid, so that if even if this pathway is inhibited, other pathway by which this amino acid is synthesized, that remains active, and as a result the plant will survive. Okay, or another method of creating herbicide resistant plant is by what? So there are few bacterial strains which use an alternate form of EPSP that is resistant to the glyphosate inhibition. So there are bacterial strains which are resistant to this glyphosate. So why are they resistant? Because a they have a modified version of this EPSP gene. So because of this modified version of EPSP gene, they synthesize an altered EPSP enzyme. Okay, and as a result, that particular this molecule glyphosate that cannot inhibit the modified EPSP, while the glyphosate will only that particular gene which codes for this modified version of EPSP gene, it is isolated from this agrobacterium strain CP4 and cloned into the crop plant to provide herbicide resistance. So okay, so if you generate a genetically modified crop which has a modified form of this EPSP gene, so that when you give that herbicide glyphosate, it will not impair the growth of that particular plant. Okay. So for the crop plants commercially available with herbicide resistance are soy, maize, sorghum, canola, and cotton. So other than these herbicide, there are different types of other plants as well. So high yielding varieties which are hybrid herbicide resistant, which are drought resistant, which are uh, which can give better yields of that particular crop or fruit. So main objective of production of herbicide resistant genetically modified crops is what? So what is the objective of generating such herbicide resistant GM is that your plant, that when you apply that herbicide, it does not affect your crop. So, but it will eliminate the weeds from the field without use of manual layout. So this is wrong option. Eliminate weeds from the field without the use of herbicide. Encourage eco-friendly, reduce herbicide accumulation in food particles for healthy safety. What will be the answer for this question? Answer is actually option A or D. Answer is option D. Reduce herbicide accumulation in food particles for healthy safety. for health safety. Hmm. So you do not want, so when these herbicides are applied to the crop, you do not want that herbicides to be incorporated into the plant, or when you consume those vegetables or those crops, they do not come to your food. So that is why where the purpose of production of herbicide resistant genetically modified crop is to prevent the accumulation of those chemicals in the food for health safety reasons. Okay, so golden rice is a promising transgenic crop. When released for cultivation, it will help in what? So answer is actually elevation of vitamin. What is the answer for this question? Vitamin A deficiency, isn't it? So you all have learned about read about this golden crop, isn't it? Golden rice, golden wheat, golden rice, isn't it? So basically, golden rice is a genetically engineered rice that produces beta carotene. So beta carotene is what? It is a precursor of vitamin A, which gives it a yellow-orange color. So when you have incorporated this vitamin this gene, this particular gene which codes for beta carotene into the rice, that will help in the alleviation, or it will reduce the vitamin A deficiency, and it gives a yellow color to the rice as well. Okay. And why was it developed? Because this vitamin A deficiency is a serious public or it is a serious health issue and in developing countries, and rice is a staple food. So since rice is a staple food, if we can add the genes for beta carotene synthesis, then it can be a dietary source of vitamin A and can significantly improve the health of vulnerable population. Okay. So what is the answer of this question? Practice of catching fish from natural resources. The practice of catching fish from natural resources, capture fishery. Okay. So there are two types of fishery. One is capture fishery and another is culture fishery. So when we are catching the fish from natural resource, then it is capture fishery. But when it is when it is the practice of catching from a culture, when you are growing catching a fish from that particular culture, then it is culture fishery. Okay. So there can be for when you are culturing a particular type of fish, or that type of fishery is called cultured fishery. Okay. So captured fishery refers to the harvesting of naturally occurring fish and other aquatic organisms directly from their natural environments, such as ocean. So from the natural environments, when the fish is captured, such as it can be oceans, it can be rivers, lakes, reservoirs, without any human intervention in their breeding or rearing. There is no artificial breeding or rearing. So that is captured fishery. So fishes are caught directly from the wild using various methods like nets, lines, or traps. And populations are not artificially stocked or managed for growth. So this is different from culture fishery. Culture fishery is what? When the fish are bred, raised, and harvested in controlled environment. Okay. So capture fishery in controlled environment means you are providing an artificial environment, and you are uh, allowing that particular condition so that particular fish can grow, okay, or breed. The capture fishery can be when you are capturing that fish from sea or ocean, then it is marine. It can be marine, it can be inland. When the fish is captured from the rivers, lakes, and reservoirs, it can be artisanal, industrial, or recreational, depending on the skill and purpose. Now, animals are selected for breeding on the basis of all of the following except what? So this is an easy question. So you can will select animals for breeding on the basis of their memory system, and other for cows or for buffaloes. The cows or buffaloes which can produce more milk. Only those type of cows will be selected for breeding. Ratio of yield to diet, or so an animal which will give you more yield but will require less diet. So that type of animals will be selected for breeding, or having long ears. So the having long ears is not a criteria for selecting animals for breeding. And also you can select animals for disease resistance. So option A, B, C, D, A, B, D. These are all the features that you look for in an animal when you are selecting it for breeding. But option C, having long ears or any physical or these ears or eyes. So these are not criteria for selection for breeding. Okay. What is this answer? So answer is for this already given. The breeding used to develop pure line in any animal is what? So what is meant by pure line? Pure line means that particular organism is homozygous for that particular character. Uh, that is so develop pure line. Pure line variety means what? When that particular. So basically, you know about Mendel's genetics. So it is homozygous means that it has both the copies. The both the copies of that gene will be will code for that for that particular character. So if you are selecting for an animal, say for example, it is that animal is able to produce very high yield of milk. Then the gene which is responsible for the synthesis of that particular character, or for giving that particular, the both of the copies of those genes will be the same. Okay, so when there is a mating between that particular animal and another or inbreeding between those particular animal, then it will always give an animal which is having that particular character. Okay. So basically, when if you remember Mendel's genetics, so if you cross small a with small a, then that will always give you small a. It will never give you capital A small a. So so that is inbreeding. So you can only develop pure line by inbreeding. By inbreeding means that particular animal is made to breed with an animal having having another animal having the same desired character multiple times. So when you are breeding that animal within the same of the same breed, then it will help in the development of pure line. Okay. So now coming to the plant tissue culture. So in plant tissue culture, there are these 8 different steps. So you take a potted plant. So from a plant, if you take any part of the plant, it can be either a root or a stem or a leaf, part of the leaf. So that is called explant. Then you sterilize that particular portion of the plant, and then you inoculate that explant in the sterilized media. So once you inoculate that explant in the sterilized media, so the media will have different components. It will have vitamins, hormones, these nutrients, and that particular will growth conditions will allow the explant to develop into callus. So callus is an undifferentiated form of the plant, and then from that callus, by treatment with a particular ratio of the auxin cytokinin, there can be generation of roots and shoot, and after that you get the plantlet. That plantlet is potted into the soil, and then it is it is grown in different conditions in different environments, and so that is called acclimatization, and then the plant is transferred to the field. Okay, so there are two terms which are usually discussed in plant tissue culture. One is callus and another is explant. So explant is any part of the plant. As I was discussing, it can be a root or a shoot or a leaf or a stem, part of a stem. So that is explant. So that is the starting material for in vitro culture, and it can be derived from various part of the plant depending on the experimental goal. And what is a callus? Callus is the differentiated form of the explant. So it is a mass of undifferentiated unorganized plant cell that forms from an explant. And explant, when it is cultured under appropriate conditions, usually with specific plant hormones. So for development of a callus from the explant, you require hormones, specific plant hormones, so that you get an undifferentiated mass of tissue. So the callus is not a natural plant structure, but is induced in vitro. So that is something that you get when you are doing plant tissue culture in vitro conditions, and can later be manipulated to regenerate into whole plants or specific organs. So there are what are the procedures involved? First, you will require the culture media. The culture media must be sterilized. You require the explant. Then after you get the explant, then you will have to inoculate that explant in a but in the culture which have autoclaved and which have sterilized. After inoculation, you have to allow that the explant to develop into the callus. Okay, into the callus. So that is callus formation. The next step is what? Incubation for. So after the callus formation, you have to treat it with hormones so that that callus develops into a plantlet. It will develop roots and shoots. And after that, once you get the that plantlet, you will grow it in different condition and allow the plant to acclimatize in the natural conditions, not in the in vitro conditions, because in the in vitro conditions they are giving all the favorable nutrients and environment that is required for the growth of the plant. So these are the different ms media, and the which are the culture media will have different composition. It will have different salts. It will have different so these grow this sucrose. It will have sucrose and these different salts which will be required for the development. So these are the different. So this is an autoclave machine where you have to you need to first sterilize the explant. So how will you sterilize the explant? And how you will also have to sterilize the culture media as well. So first you have to, for the inoculation of the explant, you need to sterilize the culture media. Then you need to sterilize the explant, because the any part of the plant you take that can have different contamination or different bacteria or any pathogen can be growing on it. So you need to treat it with different compound or different chemicals like Tween 20, or these are the different ways you can sterilize the explant. Okay. Now, once you get the explant, then you inoculate that explant in the culture media and allow the callus to form. So once you get the callus, you can see in this figure that this is the differentiated mass of tissue, which is called callus. Then after the callus is formed, that from that callus you will get a plantlet by induction with this cytokinin, auxin, and these different hormones. So once you get the plantlet, you grow it in different environments and allow it the plant to acclimatize. Okay. So what are the pair of hormones required for callus differentiation? The answer is auxin and cytokinin. Okay. Why is it auxin and cytokinin? The answer is auxin, because auxin promotes the growth of root and cytokinin promotes the growth of shoot. Okay. So auxin is this. There are synthetic auxins, indole acetic acid. Then there is cytokinin. So you can control the specific ratio of these two hormones in the culture media and determine whether the callus will differentiate into root, shoot, or remain undifferentiated. So high auxin to cytokinin will promote root formation. Low auxin to cytokinin. So if the cytokinin is more, it will result in shoot formation. If the auxin is more, it will result in root formation. Answer is option B. The callus is generated into complete plantlets by altering the concentration of what? By altering the concentration of hormones. So this is the same question. So if you alter the concentration of auxin and cytokinin, then the callus can be. So there has to be a particular ratio of this auxin and cytokinin. So when an so first you will develop the root, then you will form the shoot. So first you give a high auxin concentration so that the root is developed. Then after that you give a high cytokinin concentration so that the shoot is developed. Okay. But if you keep both the concentrations same, then at that point the callus will form. Neither the root will form nor the shoot will form. So that state is called that particular tissue that is formed, that is called callus. The answer is option B. If PCR is used to introduce random mutations rather than specific mutations, it is known as what? It is known as error-prone PCR. So that is a way where you can produce random mutations in a protein by error-prone PCR. So but there can be multiple mutations, and that will give you different variants of the or different mutants of the protein, so that next when you get different mutants of the protein, then you can screen, or for example you want to um, have a therapeutic protein, or you want to have a protein which can be used in some industry, say in a detergent industry. So you want or you want a thermostable protein for any application. So then you want different mutations, and then you will screen that which mutant protein is giving you is more thermostable, or which uh, particular protein is so that is when error-prone PCR is used. The answer is option B. Small chemical groups on the antigen molecule that can react with antibody is what? So those are the epitope. So epitope are different, is a part of the is a part of an antigen. Okay, so like your spike protein is an epitope, with so an antigen can have multiple epitopes. Okay, this option. So that is was all for this extra session. So these are the applications of genetic engineering. This part is not very difficult to understand, and I hope the questions we discussed in this session were clear. Is there any more doubts? So if there are no more doubt, then we will meet again on Monday, and that will be the last session for this particular course. If you have any doubts, then we can discuss those concepts, and also we will cover some of the points which are covered in week 12. Okay, that was all for today. Thank you for your attention and today's session.